Hi there, Dr. Bill.
Hey there, Alan. What happened? Hmm?
There's Dr. Clayton.
No. What are we doing to you?
Hello, Dr. Clayton. I think you're muted, Dr. Clayton.
We should now have both sound and vision.
Yes, sir. Good to see
Excellent.
you. Good to see you again.
Likewise, likewise.
Thank you for being with us, Dr. Clayton.
William, it's a pleasure. William, or do you go by Bill?
Bill, or hey you, or
Okay.
right. So where are you? Where are you? Uh, where
Where am I coming from?
where are you?
People often ask me that. I
Where are
wrote
you?
today, Melbourne in Florida.
Oh, okay. I was going to say in Australia. It's the next day, right? Or the day
No,
before.
no, no, no. I'm down amongst the gators.
Okay. So one of our is that where you live?
Yes, yes, it is.
Okay. So one of our good members is there. Stefan Hartman.
Uh, I
He's,
don't. I have to admit that I don't.
he's one of our superstars. So and he's in Melbourne. So
Oh, well, that'd be interesting. I mean, if he's coming on to this meeting,
he's
if
usually
we
here
have
for
a
a while.
chance.
Yeah. So
Excellent. Well, perhaps we'll have an opportunity to meet.
so we've been doing this meeting since 2020. Uh, we've been here just every Tuesday, except for two. The end of 2024, Tuesday, Christmas Eve and New Year's Eve were Tuesday, and I took off. But we've been doing it every week. And we usually end up with anywhere from depending. Lately, it's been a little bit less, 35 to maybe 80. We get pretty regular. So
Okay.
usually we have a
Well,
few,
it's
few more by now, though. I'm a little bit worried here. It's 5 of 5, and so but
well, I
they
see we
filter
have
they filter in
six
and out
people.
all the time. So
Hmm.
so usually what we do, one of our members is taking Andrew Wiles' integrative medicine fellowship. So he usually gives us a 5-minute rundown on what they've learned that week. So the rest of us don't have to do that. So, and then we introduce you.
Okay.
Okay, it's kind of how we
Yep,
do it.
that sounds fine.
Or you can introduce yourself.
Look, I'll just go by the house rules. Whatever you're comfortable with.
Yeah. Okay.
I'm a little surprised you took Christmas Day off, though. I mean, I
I
gotta
know
say
it
that's
was Christmas Eve, not Christmas Day.
slight.
By Christmas Day, it's like I'm ready to go do something, But it was snowing here Christmas Eve, so
And we don't get much of that
sort
down
sort
here.
of sort of sort of stayed in. Yeah, I know.
Hmm.
Although I was in Miami but 3 weeks ago. It was the
Hmm.
coldest week of the year that they've had in 20 years there. It probably was cold where you were too.
Oh, it was. It's killed off a lot of the vegetation. A
Yeah,
lot, the
the
plants
geckos
down
were
here.
falling from the trees.
Yes, they were, but a lot of the plants down here are not cold resistant. So there won't be many mangoes this year.
All right, Dr. Nario, there you go. You can cash in on the mango crisis.
Where is Mango going?
He's in Melbourne, Florida, with a friend of ours, right? You know who
Oh,
lives you know who lives there, right?
Melbourne, Florida.
Yeah.
Oh, Marlene.
No, Marlene's are near Tampa.
Who's in Melbourne?
Stefan.
Ah, okay. So, Dr. Clayton, you know Stefan?
He says he didn't.
No, well, no, this is a big country. There's a lot of qualified healthcare workers floating around, and I can't say I know all of them.
Yeah, he'll probably be here. It'll be nice to introduce both of you guys so you can connect.
I should point out that I am an alien. I'm a legal alien, but I'm not from your country. I'm not from the United States.
Oh, where are you from?
From Mars.
Um, well, I've been accused of that. It's a small, damp, wet island in the middle of the Atlantic, where men wear skirts and we dance on swords and that kind of stuff.
Huh. I think Dr. Bill wants to be in
I think Dr. Halassa would like
that
that.
party.
No, it's by invitation only. I hate to disappoint you.
Well, he's gonna invite you again. He's gonna make you fall into that trap. He has this move that he does to make people come back here.
Okay.
So watch out. Well, I'll be in Florida in next month, so but it's Clearwater though. Dr. Bill will be in Miami, right?
I'm going to Miami. Yeah, I'm going to Miami next week too. I'm
Oh,
going to-
back to back to back.
yeah, two. And I was in Miami at the end of January when it was freezing cold. So when my grandkids were there.
Oh, well, I'm gonna be there for the H4M Spring Congress, so if you're gonna be there, Dr. Clayton, um, hopefully we can see each other.
No,
Oh, absolutely.
I'll be at the AMMG.
Oh, is
Well,
he AMC?
I tend to move around a lot. In the next few weeks, I'll be
Sure.
in
No,
various
I'll have
venues in Europe.
No, I mean, just send me the link again. For some reason.
Okay. So, all right, just give me 1 second. We'll get started here.
Afternoon, everybody.
Hi there.
Hey, Austin, how's it going, bud? Hey, Bill, or
Another
hey,
sunny
Al.
day in Northern California.
I'm enjoying it with you.
I had to send a link to somebody, so. Okay. Okay. There's that Aaron Marin Uchi. Right?
How's it going, Doc?
It's going.
Thanks for having us and allowing the doctor to come and present this.
Yeah.
Really appreciate it.
Yeah. Can I toot your horn a little bit here for a second?
Yeah, would love to. Thank you.
So just everybody. So Aaron is he's my peptide broker. So he negotiates from same places I used to buy them a lot better prices. So if you're
Thank you,
interested in one place to get everything, we'll get you his contact information.
And don't forget the stem cells. We do the
And don't forget the
Morton's Deli and exosomes. Yeah.
Yeah. They do exosomes. They do all of that.
And now the omegas. We're in. Let's go. We want to help your
Yeah,
help
they do
your
all of that
patients.
weirdo stuff. So
Thank you, Doc.
so okay, well, I guess it's a little after 5. So I don't know, Dr. Nario, our census has been going down a little bit here. What are we going to do to whip up some interest here?
Yeah, we need to get them going in again. We need to get that CME going, huh?
I don't know if that did it or not. The guys that were on here regularly, did anybody use the CME? Did you? I did it. I did it a couple of times when we first got them, and then I didn't bother after that. So
Well, you said it's down anyway.
I haven't even heard from them.
I know we're going to be getting some of the homeopathics in here because me and Dr. Quinn are kind of
Okay.
rallying up the troops.
All right. Don't despair. But in the meantime, why don't you let us know what's going on over at the Andrew Wiles School of whatever it is that the hell you're doing there.
All the shenanigans. So today our tip of the week basically is related to it's a peptide. and I'm getting a lot of, I guess, feedback from patients also about this and clinicians, and also, of course, the school chimes in. So most clinicians first hear about GHK copper, so the copper binding tripeptide discovered by Lauren Picard, Dr. Lauren Picard, in the context of aesthetics, so skin creams, hair serums, and things that promise to make us look like we slept 8 hours and drank enough water. But the newer literature suggests GHK copper may be doing a lot more than helping dermatologists and sell expensive moisturizers. GHK is actually a small peptide composed of glycine, histidine, lysine. So that binds copper and acts as a gene regulating signaling molecule. So genomic analysis suggests it influences thousands of genes involved in inflammation, antioxidant defense, and tissue repair. So you would notice it's not just for the face, for aesthetics, and interestingly, circulating levels decline with age, which may explain why when we're 25, we bounce back from everything, and by 50, we pull a muscle reaching for the remote. Now, some of the newer studies are expanding our view of what this peptide might do. So, for example, a 2025 study in Frontiers in Pharmacology looked at GHK copper in a mouse model of ulcerative colitis. The peptide promoted intestinal mucosal healing, strengthened tight junction proteins, and even reduced inflammatory signaling through SIRT1 and STAT3 pathways. And also now, before anyone starts prescribing this for IBD tomorrow morning, it's still an animal study. It does not suggest interesting gut barrier anti-inflammatory potential, and it does kind of promote that. But again, initial studies are being presented to us. Another one in pulmonary medicine. So we've been seeing GHKU copper starting to do some cameos. And the 2024 paper in Redox Biology showed GHK copper reduced lung inflammation and fibrosis in silicosis models, largely by decreasing oxidative stress via peroxidoxin 6 pathway. So can that be applicable to other lung pathologies? Maybe, but again, that's not what I guess we're pertaining to. But in the future, that might be a standpoint. So apparently this tiny peptide is not only helping skin, it may also be telling fibrotic tissue to calm down, as you can see. Neurologically, also 2024 Alzheimer's model study found intranasal GHK improved cognition, reduced amyloid protein burden, and lowered neuroinflammation in mice, which raises the interesting possibility that the same molecule used in anti-aging skin serums might also have neuroprotective signaling effects. Another 2024 aging study, GHK copper, could reverse age-related fibrosis, modulating myofibroblast activity and promoting apoptosis in senescent cells. So in other words, it may help tissues stop behaving like grumpy retirees and start acting a little younger again. So there's also mechanistic data showing GHK copper can prevent copper and zinc-induced protein aggregation in neuronal models. And this is relevant to metal-related neurotoxicity. And in the dermatology, so this is where it really shines, a 2024 clinical study showed topical copper tripeptide improving seborrheic dermatitis and scalp inflammation. And this is actually more useful, as we all know, in cream form, injectable, maybe, and even oral, even less because of gastric acid destruction. So finally, biomaterials research shows GHK copper can enhance antibacterial activity and accelerate wound healing, so improving collagen deposition while reducing inflammatory cytokines. So when you step back and look at the literature, GHK copper may not simply be a cosmetic peptide. It's increasingly looking like a broad regenerative signaling molecule involved in inflammation control, fibrosis regulation, tissue repair, and potentially aging biology. Of course, we still need to do larger human clinical trials, but the trajectory of research suggests that GHK copper may eventually become part of a new class of bioregulatory therapies in regenerative medicine. If it also helps our patients' skin while doing that, well, I doubt anyone will complain. So that's our leak of the week.
All right.
So, Dr. Bill, I think you're using this orally, right? You have this in your.
We have it as injectable, and we do have it orally. Yes. So you're suggesting it as a cream?
Well, the studies are actually more for cream form. So this is really more for wrinkles, anti-aging, and just to remind everybody, injectables, they sting. They have a little pain in them.
They do.
So,
That's
right.
That's the reason to use them. That way you know that they're working.
Yeah, that's also a good sign for that.
I have A topical for hair growth.
Yeah, that's the one. But they also inject that on like hair follicles. So there are such methods out there.
Yeah,
So
we do that, Doc. We actually have an exosome that we use with the GHK copper and biotin, and we microneedle it, our doctors microneedle it on the scalp, and we
right.
actually guarantee our outcomes because it works so well.
See, I'm tooting your horn too. I didn't even know about it.
Thanks, Dr. Nario. I know you and I we've connected once. We need to connect again.
Yeah, of course.
There you go. All right. So the cream comes in what kind of concentrations?
So dosing wise, I think the cream. Let me remember the dosing. I'll type it in the chat for you. I forgot the dose exactly. I don't know if Aaron knows the dose.
I don't off the top of my head, not for the concentration that in a cream.
Yeah, but cream. So that's the one that where GHK copper studies have been backing up with. So it's really effective, and I've seen this in patients. They rub it on their skin for anti-wrinkles. So that's where it
You
really
want?
shines
See,
the best.
there you go.
Yeah, they use it in the HA serums as well.
Right. So there's also liposomal forms of this, so orally, so they can divert themselves out of that gastric acid pathway. And there's a company that sells that. Everybody knows this company. So yeah, and yeah, that's a better form if you're going to go for oral versions of this.
Um, One of the
Hey,
big
Melvin,
peptide companies shut down this week, didn't they?
Which one?
Peptide Sciences is shut down.
Yeah, Peptide Science did shut down this week, Doc.
Are you sure it's just not the website?
No, it's not the whole compound. Yeah, but they did shut the website down. It's Visa, MasterCard do not like peptide companies. Um, and so they're having trouble with the processor, which I'm going to try to help them out with since I do that as well.
All right. Here's the dose that you're looking for. So it's actually 5 milligrams, and then also 1 ml in terms of the pump and spray. So these are I mean, this is for the company that does this is, I was talking about liposomal, this Quicksilver. So that's one that I guess has been vetted for that it has a higher bioavailability.
Well, Carol Peterson will say you better use them or else, right?
She'll give us samples when we see her in A4M.
She'll give you samples.
Yeah.
Okay, thank you. All right, everybody. So today we have a very special guest, Dr. Paul Clayton, summa cum laude in medical pharmacology from Edinburgh University. Former chair of the Forum on Food and Health in the UK, senior scientific advisor to the UK Government Committee on the Safety of Medicines. Dr. Clayton was a fellow at the Institute of Food, Brain, and Behavior in Oxford. Dr. Clayton works with leading doctors and clinical scientists at centers of clinical expertise in many countries, designing and supervising preclinical and clinical trials of pharmaconutritional interventions. His books and ebooks include Health Defense, After Atkins, Natural Defenses, Out of the Fire, Let Your Food Be Your Pharmaco, Nutrition, and Strengthening Your Immune System. So we are honored to have you, Dr. Clayton and Alan. Thank you for bringing him along, and thank you for sponsoring us at our conference and being with us, for the last few months. So
you're very welcome. Thank you.
With that, if I got anything wrong, Dr. Clayton, please correct me and take it away. It's yours.
No, thank you, Bill. That was broadly correct. I'll go with it. First of all, thank you for the opportunity of speaking with you. I'm hoping that at the end of this, there'll be an opportunity for some questions and answers, and the more aggressive the questions, the more I like them. I'm a scientist really more than a clinician, and it's from these interchanges of opinion that we're able to modify our own models, and we know that they do need modification. So without further ado, let me share my screen, and I'll start off by showing you a few slides. Let's see if I can get this to work. With any luck, you should have a slide on your screen by now. So what are you seeing right now, folks?
We're seeing Zoom, a Zoom screen, and now we see a countryside. There we go.
Oh, Lord, that's not the point at all. Do you now see a PowerPoint slide?
Yes, it says the end of medicine.
The end of medicine.
The end of medicine is pretty much what it is, which might sound a little apocalyptic, but there is a purpose to this, point to it. The current model of healthcare that we use is primarily pharmaceutical. It's pharmacocentric, let me put it like that. And it's not really doing us very much good from a public health perspective. And I'm going to show you some figures here to demonstrate which direction we're moving in. And the delta is entirely negative. The situation in North America, and particularly in the United States, is quite revealing. And one of the reasons for that is that you have been running national health surveys since 1935. The data is not perfect, but it's good enough. And what we know is that back in the day, in 1935, when you add up all those people who were presenting with a single chronic disease condition, it was about 10% of adults. Amongst children and adolescents, 2%. Now that's a very conservative figure. It's probably closer to 1. But I have to say again, the data is a little diffuse. Now, if we move closer to the present time, by 2000, you now see a rather different picture with 45%, almost 1 in 2 adults presenting with a chronic condition. By 2020, that's increased to 60%. Amongst children and adolescents, the figures have risen from 1 to 2% to a staggering 40+%. This is an entirely negative direction. And because more people are presenting with chronic conditions, you see more individual cases with more than one. In 1935, less than 5% of adults had more than one chronic degenerative disease. By 2018, that was 27.5%. By 2022, that was 40%. We can't really continue down this road. It's an incredible burden. It's one of the reasons why healthcare systems everywhere are increasingly going in for rationing schemes, why I think we have to start to think more about moving to a preventative approach to public health if we could come up with one that worked. Those are the same results in graphic form. And if you do a projection into the future by about 2080, what that means is if we carry on, it's going to be somewhere like 100%. Just about everybody is going to have one or more chronic degenerative condition. Now, I know that during this period, there has been a proliferation of pharmaceuticals. The pharmaceutical industry have grown fat by that one metric, at least profits. They've done extremely well. But in terms of public health, they have failed us utterly and completely. And one of the reasons for that, of course, is that most pharmaceuticals are simply too toxic to be used preventatively. And we can't identify the people who are most at risk anyway. And all the multiple SNP analysis studies have confirmed that. It's nothing to do with spending. I don't think we're spending our money wisely at all. You can see the way in which healthcare spending in the United States has increased from $500 billion in 1970 to $5.5 trillion in 2026. That's based on constant 2024 dollars. So that's a real trend there. During that time of increased spending, healthy life expectancy at birth has fallen. Everything's wrong. Nothing matches here by 1.41 years. And we see the same trends in the United Kingdom. That's my base point. Males' life expectancy has fallen between 2011 and 2021 by a shocking 2 years, by females 3 years. This is the lowest healthy life expectancy since records began in 2011. We're going backwards, as are you. We know that there is a strong socioeconomic gradient. So Richmond is pretty much at the top, with people earning in the top 5%. Tedville, bottom 5%, and you can see the difference in healthy life expectancy in males and in females. It's very considerable. And this is merely confirming to us the picture that is already well established, that poor diet is one of the most important determinants of early death, which means fundamentally preventative death. So what's increasing? Really, the question is what isn't increasing? These are a list of some of the diseases that are increasing in frequency in the general population. They're also decreasing in latency. So we see many of them emerging in progressively younger groups of patients. apart from the neurodevelopmental disorders, which are happening at probably in the same kind of age range, but we're seeing more of those. The more perceptive amongst you have noticed, always, obviously, this is an alphabetical list. So here are a few more, and this list is not complete. Almost every week, people come to me with new data saying, here is another condition that's increasing. There's only one that we know of, one chronic degenerative disease that is decreasing, and I won't reveal it to you now. If you're are interested in that, we can have perhaps talk about that at the end of this. Now, because so many people are developing these conditions, and because so many of them have long latency periods, you have these long preclinical phases. When we look in detail at people who mistakenly believe themselves to be healthy young adults, we find about a fifth have quite advanced liver disease. They've moved through MASLD, they're now in MASH. About a quarter of young healthy adults are moving towards osteoporosis. At least a third of healthy young to middle-aged adults are developing hypertension, atheromatous disease. They're at risk of thromboembolic episodes. We know that about a fifth of healthy middle-aged adults are showing signs of ARCD. They're moving towards dementia. And perhaps most worryingly of all, this is a really alarming finding. In the age range 40 to 49, almost half of all adults are demonstrating pre-frailty, which is the preclinical precursor to sarcopenia. And sarcopenia is increasing, of course. It increases the risk of all-cause mortality, just about everything. This makes everybody extremely vulnerable. Now, you might think that the deposition of atheroma in your coronary arteries, or the thinning of bone, or the progressive loss of brain cells, you might think of these as intrinsic signs of the aging process. They're clearly not. When we look at those people who live in vestigial communities, the most documented are the Hadza in Tanzania. They're hunter-gatherers, and then you have the Tsimane in Bolivia. They're forager horticulturalists, I guess is the best way of describing them. When you look at those, you see that in young to middle-aged adults, you don't see early signs of coronary artery disease. There's no fatty streaks in their arteries at all. As they get older, they don't become insulin insensitive. Their blood pressure does not rise. We don't really see benign prostatic hyperplasia. This secondary phase of prostatic growth does not occur. In both men and in women, kidneys do not develop an age-related decline in efficiency. And perhaps most interestingly of all, you don't see hippocampal or cortical shrinkage with age. So as they get older, there is little, if any, incidence of any of the neurodegenerative diseases. We do see a very small number of cases, but they're actually quite exceptional. And may I just stress that this is in a population that actually has a life expectancy not very different from ours, and a healthy life expectancy, if you strip out infectious and parasitic diseases, that is actually better than ours. So it's clear, I think, that what we have thought of previously as intrinsic signs of aging, they're not intrinsic at all, they're extrinsic. They're probably not signs of aging at all, but they are, in fact, signs of progressive and chronic intoxication. And I'll go into that in more detail in a minute. But for all these reasons, I think this increase in the frequency of disease, its decrease in latency, some of these conditions are getting more publicity than others. The increasing numbers of very young adults presenting with cancers, a whole range of cancers, is a particular concern. But I don't think we can continue to go down this road. We have to, I think, pause and consider how we got to this point. Now, this is a very, very interesting slide. It took me 2 years to pin it down. It's hidden away in the bowels of the NIH, and it was generated by the Division of Vital Statistics at the National Center for Health Sciences, which no longer exists. It has become wrapped up into what it is, the current NIH. And this particular graph shows deaths from infectious and parasitic diseases and other lung diseases on the respiratory tract. And the important point here, look at the solid lines. Basically, those are deaths from infectious and parasitic diseases, as you can see, including pneumonia and influenza, but a number of other respiratory conditions as well. And if you look at the dotted lines, those are non-communicable respiratory tract diseases such as COPD. Some of you, I think, refer to as COLD. lung cancers, and the like. And they start to cross over in the early 1950s. And from then on, it's the chronic degenerative diseases that become progressively more significant at the public health level. So I think it's fair to ask the question, what happened in the early 1950s to bring about this change? Now, in terms of what I just said in the previous slides about the long latency, the long preclinical phases of these types of conditions, it actually makes more sense to ask what happened in the early 40s. Let's go 10 years into the past, what happened at that period. And that, of course, was the introduction of what people call variously fast foods, brown foods, junk foods, industrial foods, or ultra-processed foods. That's when our overall nutritional status really starts to take a nosedive. And this is reasonably widely accepted. This is from IMHE, the US Institute of Health Metrics and Evaluation. They reckon that poor diet is the biggest cause of early death. And that's not just a United States phenomenon, it is a global one. We could try, I suppose, if you wanted to analyze the molecules that we put onto our plates and into our stomachs. I think that's probably not going to be a very effective way. I don't think that's the right question at all, because if you count up the number of molecules in an average person's diet over the course of a week, it may be anything from 40 to 90,000 different molecules. And each of those, almost all of those, have a pharmacological or at least a physiological effect. So there is another way of looking at this, and that is to try to determine whether behind this noise, this curtain of tens of thousands of variables, one could identify a smaller number of pathoetiological drivers, which between them link the industrial diet to the emerging pathologies that I listed previously. We know that the industrial diet affects the epigenome, the metabolome, but we can start to be a little bit more specific. We can break it down into 4 major drivers of disease. Now, I'm sure many of you can name a few others, but these appear to be the 4 major ones: chronic inflammatory stress, which everybody knows about, dysbiosis, type B malnutrition, and I'll define that term subsequently, and glycemic mismatch, which is a situation where you're pouring more glucose into the blood than you're taking out. And that's exacerbated by a low energy lifestyle. We're eating relatively little compared to our great-great-grandparents, because we have internal combustion engines and information technology. They didn't. They did their work with their backs and with their hands. They consumed more calories. And we're also eating a lot of ultra-processed foods, which have a lot of empty calories. So that accentuates type B malnutrition. And because we're so physically inactive, and it's the muscles that are the most important glucose sink, the liver has only a very limited ability to abstract glucose from the bloodstream. And that's compounded by this low energy lifestyle and this appalling diet that many of us are now eating. Makes those 2 things worse. A grim picture, but I think what this means is that at last we can begin to consider rational antidotes. We can consider ways of neutralizing these particular drivers of disease. And if we could do that successfully, we would expect to see improvements not only at the public health level, but in individual cases. And in fact, we do routinely see just this. So let's take them in turn. Chronic inflammatory stress. Most of you are probably pretty familiar with most of the mechanisms listed here. This is a very simplified diagram. And I'm just going to make the point that we need to be able to mount an inflammatory response. Obviously, an acute inflammatory response is essential in all kinds of situations. But if you have too much inflammation and it goes on for too long, it's not good for you at all. We got to be in the Goldilocks zone. We have to have the ability to create as much inflammatory sequences as are necessary to keep us free from infection, to be able to repair trauma, for example. But from the public health perspective, there is clearly too much inflammatory stress at the population and at the individual level. Now, some of that, not all of it, is down to your omega-6 and omega-3 ratio. What we're finding is, and we have a database of about 1.5 million samples now, that when you bring the 6-to-3 ratio down to 5.1 or less, that's when chronic inflammatory stops, but you still have the ability to maintain that critical acute inflammatory response. Historical ratios, from the best of the data that we have access to, they are somewhere between maybe 2 to 4 to 1. Optimal ratios 5 to 1, but where are we today? Europe is at 15 to 1. Eastern Europe and North America are at between 25 and 45 to 1. So That doesn't seem like a good thing. It's far away from the historical norm. We know that the sixes produce a range of mediators which are broadly pro-inflammatory, the threes broadly anti-inflammatory. And we know that when you lower the 6-3 ratio in a European or North American subject, what we see is very consistently, as I said, in hundreds of thousands of subjects now, is a reduction of inflammation. And we see that in mice too, as well as in men. And please excuse me, that's just for alliterative purposes. Of course, that includes women. Here's one of the issues. Some of you, I think, have seen this slide before. There's been an extraordinary increase in the 6-3 ratio in the diet and therefore in our cell membranes, partly to do with techniques that were developed to stabilize omega-6s at the beginning of the 20th century so that these oils, which could be turned into fats, and which were increasingly incorporated into the ultra-processed foods we were consuming more and more of, led to this inexorable increase in the 6-3 ratio. So what does that mean? Well, the omega-3s and 6s have got different roles. They have structural roles. They're built into the phosphatidyl phospholipids that make up the bulk of the cell membranes. But they also are the starting point for a series of messenger substances which determine how you respond to various inflammatory challenges. Some of those may be external, some of them may be internal. If there's time, I'll go into that. So there you've got your cell membrane, the cartoon version, your lipid bilayer running across the top of the slide, an inflammatory challenge. It could be a microbe or it could be a trauma. You need to be able to develop an acute inflammatory response to that, which is all about finishing off the microbe if you can, or removing some of the damaged tissue so that healing can take place. And arachidonic acid is very important here. It's producing pro-inflammatory mediators. Once the threat has been neutralized, you need to switch off that inflammatory response. And we do that starting with the omega-3s, EPA and DHA in the cell membranes, and they will stop the inflammation, bring it to a close, and at that point, you're back to where you started before the challenge occurred. Part of the problem is that the parent fatty acids, that's LA and ALA, are metabolized by the same group of enzymes. And so if you're eating a diet where the omega-6s, where LA is in excess of 4-to-1 over the omega-3 ALA, your desaturation and elongation enzymes have a preference for the ALA of about 4-to-1. Once you override that, basically you are no longer able to produce the long chain omega-3 polyunsaturated fatty acids. And because we're consuming so much seed oils now, either directly in cooking or indirectly concealed in ultra-processed foods, that pathway on the right has been choked off. And the only way to get around it is by supplementing with the long-chain polyunsaturated omega-3s directly. And of course, you can do this by consuming oily fish, which some people are prepared to do, many aren't, or you can take an omega-3 supplement. This is the problem with the omega-3s, but I'm now going to talk about polyphenols because they're involved in this inflammatory cascade as well. Many of the health agencies suggest we should be eating 5 fruits and vegetables a day. They're not the only sources of polyphenols, but they're important. The reality is actually fewer than 3 portions a day on average. A little bit of a historical context here. The mid-Victorians, that's the English between 1850 and 1900, were consuming 10 portions of fruits a day. And those were heritage strains of fruits and vegetables, which contained up to 3 times the amounts of polyphenols that today's fruits and veg do. And again, it's an interesting why that should be. If there's time, maybe we can go into that. So polyphenol intakes have fallen by 60 to 90%. The 6-to-3 ratios have been degraded as well. Why is that important? Because if you break the inflammatory cascade down now, again, in a very simplified version, this could be, let's say, an immune cell. It could be a neutrophil or a macrophage, a Kupffer cell, could be any one of those, a Langerhans cell. There's a challenge, and the lipid mediators derived from your 6s and 3s in the cell membrane, they'll initially cause the classical signs of acute inflammatory stress, so heat, swelling, redness, pain, etc., etc. You're not causing any tissue damage at this point, but this is just the first stage in the sequence. Those cells will be shedding exosomes, I think one of the members of the audience was talking about those a moment ago. And those exosomes are being shed into the extracellular space, and they contain all kinds of information, the various bits of genetic information, which are really interesting, but they also contain proteases and matrix metalloproteases, which are extraordinarily destructive because they basically tear holes in your extracellular matrix. So a healthy diet, which allows you to mount an acute inflammatory response, but does not allow you to develop chronic inflammatory stress, will not only give you a well-configured 6-to-3 ratio, it'll also provide you with enough of the polyphenols. The polyphenols do lots of things. They actually inhibit the matrix metalloproteases using a direct inhibitory reaction. They bind to the divalent metal cations, which are in the active sites of these enzymes, but they also have an epigenetic effect. They downregulate the genes that express the MMPs. So they're very, very important in modifying and modulating the activity of these extraordinarily destructive enzymes. Here's the problem. The modern diet gives us a very high 6-to-3 ratio. So the first sequence is overheating, and the modern diet also strips away the polyphenols. So the second phase is overheating as well. And now in many tissues in the body, your immune cells and some other cellular species are drip feeding matrix metalloproteases into the extracellular space, into your tissues, which could be in your joints, in your bones, in your blood vessels, or inside your skull. And they are dissolving the extracellular matrix. And again, I'm not quite sure what all of your backgrounds are. If I was talking to a lay audience, I'd make the point that yes, collagen is one of these. It's one of, there's a dozen or so collagens, but there's over 300 different microfibers. This is an extraordinarily complex tissue. And they all have different types of structures. It is, in fact, a spatially configured series of growth factor reservoirs, amongst other things. But the point is, the matrix metalloproteases chew through each and every one of these single fibers. These are the same enzymes that are released in large amounts in a relatively acute timeline by the so-called flesh-eating bacteria. And if you're unlucky enough to be infected by these bacterial types and you don't get treatment in time, basically you dissolve. These enzymes break down and dissolve bone, cartilage, fascia, skin, muscle, extraordinarily destructive. And it is these same enzymes, when they're released in small amounts over decades, lead to the progressive destruction, excuse me, of cartilage, leading to osteoarthritis in bone, leading to osteopenia and osteoporosis in the skin, to dermatoporosis. If it's in the linings of your blood vessels, it leads to the dominance of the constricting controlling systems. So you develop essential hypertension, the progressive inflammatory lesions in the linings, in the intima of your arteries, are the starting points of atheroma, and so on and so forth. Now, who are the causes of this? Well, it is, of course, your beloved multinational food brands. These are the companies that we trust and the foods that we love. None of these are toxic in the acute sense, but if you eat large amounts of these foods over long periods of time, they will make you sick and they will kill you, because 4 of the most important categories of anti-inflammatory nutrients have been stripped away for different reasons. I'm not talking about a conspiracy here. There are reasonably valid reasons for removing all of these, but they have been replaced by a number of fundamentally pro-inflammatory compounds. And this is really to do with industrial food production. It is to do with the recipes that the food companies use to create hyperpalatability, addictive potential, if you like. So you end up with foods which food universe, which is configured in a way which creates chronic inflammatory stress. It creates dysbiosis. It creates type B malnutrition. This is why, as Imhe pointed out, these foods will make you sick and they will kill you. Now, quickly, let me go through type B malnutrition, because I have mentioned that. This is the classic textbook form. This is what I was taught at medical school, and that looks something like that. Kwashiorkor, or it could be beriberi, pellagra, scurvy. Typically, micronutrients that we don't store very well. So if you switch to a diet where they don't exist, the symptoms, a constellation, a well-known constellation of symptoms will emerge in a relatively short period of time. And so our medical ancestors were able to pick this up. But we don't see that very often. We see type B malnutrition, which is characterized by depletion, not deficiency, in many different macronutrients. And not just the vitamins and trace elements, by the way, but also in a wide range of phytonutrients and fiber types, et cetera, et cetera. And very often it's accompanied by an excess of calories. And that's what you get when you eat these kinds of food. Lots of empty calories and very, very low on many of the micro and phytonutrients that we need. So this gives you type B malnutrition. A lot of people give their kids confectionery for breakfast. These are not a good idea at all. Many of us will eat these kinds of foods because we like them. They're hyperpalatable, but they're not a good idea to eat too many of them. And this is perhaps one of the most extreme examples. Coca-Cola, but this is a stand-in for all the sugar-sweetened beverages. They're all empty calories all the time and nothing else. And I just want to bring to your attention, if you didn't already know, that this version of Santa Claus is an artifact himself. If you know your history, his ancestor, Saint Nicholas, was a very lean and ascetic-looking figure. This is the Coca-Cola's imagination of Father Christmas, and he looks like he's been getting high on his own supply, full of empty calories. He probably has type 2 diabetes, hypertension, a lot of degenerative diseases, and not a tooth left in his head. The whole thing is made worse by the fact that we've become considerably less physically active than we used to. Let's just quickly go around the screen. That is not the correct way to walk your dog, nor is that the correct way to pull your luggage. The thing on the right is a remote control, and it's just an example of the myriad of little ways and some large ways in which our calorific expenditures have declined over the generations. We're eating empty calories. It means that most people are low in most things, and this is pretty robust data. We see something similar on our side of the Atlantic as well, which is why ultra-processed foods are associated with death, and they're associated with an increased risk of cancer, type 2 diabetes, increased risk of early death, and it's dose-related. You know, if they weren't already convinced that these were bad in theory, the fact that there is this this dose-related effect, I think, makes the whole thing rather more convincing. Increased risk of COVID. This is a 22 paper. One of the reasons probably why is that this type of diet leaves your immune system short of a few key catalytic factors, but also it means that you have dysbiosis. And as you probably know, the COVID virus has a home in the large bowel. And if you have a healthy microbiota there, it actually militates against COVID replication and spread. If we don't, and most of us have a pretty unhealthy microbiota now, it harbors the COVID virus and is COVID permissive. Reduced risk of dementia. That's another rather good 22 study. There's more that have come out since then that say the same thing. Increased immunological aging. If you eat these foods, increased inflammation and sepsis. That's actually a preclinical work. And then a group led by Esposito. This is a Franco-Italian Spanish group showing that if you eat more of these foods, all of the deep signs of biological aging are accelerated as well. A quick look at the international ratings. This is average life expectancy in different countries. And if you look at the bottom right-hand corner of the screen, that is the USA, position 53. Trace that back to 1950. At that time, the USA had the 13th highest average life expectancy in all of the countries that were then being tabulated. I know that this is not a perfect guide as to general health, but it's reasonably accurate. Look at the direction of travel. So from position 13 to position 53 in 2020, since that time, the United States has fallen a further 3 places in the international league table. We're not doing that much better. The UK, that's the pale blue track, at 2020 at position 36. We used to be position 10. We are the 51st state. We're very closely following the United States in terms of ultra-processed food consumption. One trace there is very clearly bucking the trend. That's the light green trace there, and that's Japan. They've moved from position 45, and since about 1980, 1990, they've consistently been in the top position. What did they do? Well, amongst other things, they developed a category of food called fashu, And these are foods which are designed to provide specific health benefits. They started off with compounds with added dietary fiber and probiotics and developed it from there. And now the Japanese food, the consumer market, has really cottoned onto this. And if you try and launch a new food into the Japanese market, if you don't have true functionality built into it, you probably won't do very well. So this is a huge improvement that has been seen in Japan, and that has been achieved really through dietary improvement alone. They haven't been selling more drugs, training more physicians, building more hospitals. They've simply taken care to get the nutritional baselines in a healthier configuration. Unlike the situation here, very sadly. So if you look at these young kids, they may think that they're healthy, but really they're not. These are the people who, when you look at them in detail, you find that they already have the seeds of those chronic degenerative diseases, which will over time grow in the preclinical or latent phase, but eventually 20, 30 years further on from now, surface as a clinical condition and possibly a very serious one. So let's consider Mr. Average, Mr. I don't know, Joe Sixpack or whatever. And he's eating an ultra-processed diet, which means he has a high 6-to-3 ratio, 15-to-1 or in excess of that, and really not very many polyphenols at all. He will therefore have chronic inflammatory stress, which can manifest as a wide number of disease states, ranging through the arthritides, through it could be MASLD or MASH, it could be coronary heart disease, essential hypertension, COLD, migraines, or a brain tumor. Chronic inflammatory stress is a driver in all of these conditions. But if he's eating ultra-processed foods, which are incredibly deficient in prebiotic fiber, then he now has a microbiota which is not saccharolytic, but it is predominantly proteolytic or lipolytic, which creates chronic inflammatory stress in the intestines as well. We can put most of this right, rectifying the omega-3s and the polyphenols. We can do that very effectively in every tissue in the body, with the exception of the gut, where you need to start to address the problem of dysbiosis, which we can only do using prebiotic fibers. So what are they and what do they do? I know that most of the market is all about probiotics. The reality is that EFSA and most other regulatory agencies have not awarded a single health-related claim to any probiotic species because they simply don't take. If you take a couple of billion colony-forming units and swallow them, they are out of their depth and they're hopelessly outnumbered, and they don't take because of colonization resistance. And we know, because we've measured this, we find that as long as you're taking a probiotic, we may find evidence of that probiotic strain in the stool while you're taking it. The day you stop taking the probiotics, they're gone. They simply cannot settle. However, if you eat prebiotic fibers, they feed the saccharolytic species, here marked in green, and they're producing a range of postbiotic substances which cumulatively are powerfully anti-inflammatory and also extremely chemoprotective. That is, there are compounds there that will take a wide range of cancer cells and induce either mid-cell cycle arrest. They will induce one or more forms of cancer cell death, or they will induce redifferentiation. This is a very interesting and potent package of chemoprotective effects. Take the prebiotic fibers away, the saccharolytic species starve, they become less numerous. Nature and the gut abhor a vacuum. Into that ecological space emerge increasing numbers of proteolytic and lipolytic species marked here in red. Where do they get their protein from, the proteolytic species in particular? Well, about 10% of the protein you consume will probably make it as far as the large bowel and be fermented, producing, amongst other things, ammonia, which is not necessarily a very good thing for you. But these bugs are getting their protein from other sources. They get it particularly, they like to eat mucin. So as they consume mucin, they degrade the mucus layer and they come into direct contact with the colonocytes, cells along the bottom of the screen. And because many of these bacteria are gram-negative, they're coated with lipopolysaccharides, extremely potent pro-inflammatory compounds, same molecules that were the active in Coley's toxins. For those of you who are interested in medical history, you have enormous chronic inflammatory stress. So you are moving towards IBS, IBD, and at a certain point, your tight cell junctions start to break down. You now see the translocation of toxic molecules such as LPS into the bloodstream, you have endotoxemia. Immediately, you're running into hepatic problems, cardiovascular problems, central nervous system problems, bones and lung and everything else. Dysbiosis has been linked clearly to all of those conditions. And if the tight cell junctions break down even further, you see the translocation of enteric species into the bloodstream. And this has now been identified as a potential, the important cause of what we used to think was autoimmune disease, but may well be due in a surprising number of cases to intracellular infections. Your immune system is trying to get to the microbes. They see the fragments of these bugs on the surface of your host cells. And in so doing, they wreak collateral damage. And if the infection, this intracellular infection, is in the thyroid, you might well end up with something like Graves or Hashimoto's. That seems to be the new model. And there's an interesting work being done now with a number of antibiotics which are able to achieve bacteriostatic or bactericidal concentrations inside cells. And if you give these courses for long enough, some of the autoimmune diseases, and they may not be autoimmune diseases at all, actually go away. They are cured. The remedy for this, put prebiotic fibers back into the diet, reestablish a predominantly saccharolytic microbiota. And if you do that, that has extremely positive effects. This is a WHO study, big numbers. And they found that higher fiber intakes reduce early death by 30%, cancers and cardiovascular and dementia. And you get reduced microplastic uptake as well, if that is of interest to you. This is a very potent series of benefits. It's more than you can achieve with any one drug, or in fact, all the drugs that you could have access to. And you have to look at this in terms of an evolutionary long match. If you go by the pre-Christian Mesoamerican data, and this is based on some very beautiful quantitative archaeological work, it looked as if the people at that time, the Ogham folk, for example, were consuming about 140 grams of prebiotic fibers a day from roots, shoots, and tubers, and nopales, sources like that. So they would have had a very predominantly saccharolytic microbiota, and that has a knock-on effect on the microbiota in all the other tissues as well. And they're eating about maybe a fifth of their daily calorific requirements in the form of these fibers. We don't know how healthy they were. They didn't leave medical records, but we do have medical records for the mid-Victorians, England, 1850 to 1900. They're not consuming as much, about 40 grams a day of prebiotic fibers, and different sources. Helianthus tuberosus, for example, is very important. What effect does this have on them? Well, that's an interesting population because it's extremely intensely documented. We have medical texts from the day, medical reports, lots and lots of autopsies. The doctors of the day conducted, even country doctors, 3, 4, or 5 autopsies a week, and they became very good at it. They produced better autopsy reports than many of my students can do today. And I won't go into all of the findings, but I'll simply talk about colorectal cancer because the patterns there are very glaring. The doctors diagnosed it when they found it, as I said, without prejudice. It was rare, and so rare that they could determine its occurrence within families. You can't do that anymore because the overall figures have increased so vastly. But back in the day, it was rare, and they could tell it was inherited, even although this was pre-Mendel and pre-DNA. It was very obvious. Everybody else was protected. Where are we now? We're down to 4 grams a day on average. So we have fallen from 140 to 40 to 4. And of course, many of the conditions that we think are linked to this, IBS and IBD, have increased dramatically. Colorectal cancer, huge increases. Appendiceal cancer, ditto. And there's many other conditions that appear to be involved. This is what a prebiotic fiber looks like. It's a carbohydrate, obviously, but the linkages are such that these carbohydrates cannot be digested. So they pass through the small intestine intact. They have a calorific content of only about 2 calories a gram. And this is a small molecule, so it's fermented very rapidly. When the bug gets hold of it, it's eating it like beads along a chain. These are short fibers, so the fermentation is rapid. We have to add larger amounts of inulin, same structure, but longer, so the fermentation is slower. Larger amounts of 1314 beta-glucans, larger and more complex molecules, slower fermentation, and the largest amounts of resistant starch, which you can get from sources like green banana but now the fermentation is very slow indeed. And you need to have all of these different types of fibers present because when this blend enters the large intestine, everything is moving. So the FOS starts to ferment first, switching the microbiota from proteolytic to saccharolytic in the ascending colon. And then the inulin takes over, obviously not as neatly as this. It's a kind of a relay race. And I'm just showing you this diagram to get over the idea of sequential activity. Obviously, there's more overlap. And then the 1,3,1,4 beta glucans take over. And finally, the resistant starches. And in this way, we can achieve this shift to saccharolytic species throughout the entirety of the gastrointestinal tract. And when we do this, what we find is that IBS goes away, IBD starts to resolve. And then if you have, for example, MASLD or MASH, that starts to get better as well. You can do a fiber scan readings and check it for yourself. The liver flexibility starts to return quite quickly. So there's the industrial diet and low energy lifestyle. This is what's making us sick. We can talk about the antidotes. We can suppress and prevent chronic inflammatory stress very quickly and very easily and very consistently. Not with the omega-3s on their own, they don't work. But if you combine them with the right polyphenols or lipophenols, we can switch off chronic inflammatory stress just about as effectively as a glucocorticoid. We can restore eubiosis with appropriately combined fermentable carbohydrates. We can alleviate type B malnutrition and bring the microphytonutrient profile up to something similar to you would see in a vestigial culture with a very broad spectrum support program of micro and phytonutrients. And as for glycemic mismatch, well, that's up to the patient. We tell them, obviously, to eat less sugar and starch, take more exercise. Some can't or won't do that. And for those, there are AMPK upregulators, which reproduce just about all of the metabolic effects of exercise. They're regarded as exercise mimetics. So I don't want to take up all your time. I hope there's time for questions. You can buy the books, but you don't have to. Just go to the blog. All the information is there with a proliferation of references that you can use as jumping points into the wider literature to check whether there's anything at all in behind what I'm saying. Now let me switch everything off. And hopefully there will be time for some questions.
Okay. Thank you so much. What do we have what for as questions are concerned? Chat. Okay, I don't see any questions here on our side. You got anything, Dr. Nerio?
Who is the
Yeah. So that was a great presentation, Dr. Clayton. So the thing here is when you talk about again I think pharmaconutrition is a common term that I've been hearing from you, and I'm sure you pioneered this concept. And how is this different from the conventional nutritional advice that us doctors give? Because sometimes it's so generic on our end, and as we all know, as medical providers, we didn't get much training in nutrition, so there's only limited hours that we had for school for that, and most of this will be almost self-study on our end. So how do you as you conceptualize this? How does it differ?
Well, first of all, I think you put your finger on a very acute problem. I believe that in the United States, only about 20 to 25% of the medical schools teach any nutrition at all. And in those facilities where it is taught, it might be 15 hours in a 4-hour course. And there are reasons for that, historical and economic, and perhaps one could even say political, but we don't have to get into that. So how does this differ from nutrition? I think that when most doctors give nutritional advice to individual patients, it's pretty generic. And what we say is, well, eat more fruits and vegetables and maybe cut back on the seed oils, not quite so much salt. We'll tell them things like that. And the reality is that these messages are not particularly convincing. They're not very effective. Your voices are drowned out by the advertising spend that the food multinationals saturate our children and our media with. So it's hard for us to have our voices heard. The differences between nutrition and pharmaconutrition. So I'm finally coming around to the core of your question. As I said, nutritional advice tends to be rather broad. It's not been shown to be particularly effective. What I have been trying to do is to develop very specific antidotes for the fundamental ways in which the modern diet and lifestyle is making us sick, boiling it down to really 3 programs, which can be easily combined. Each one is like the leg on a 3-legged stool. You can stop chronic inflammatory stress with this. You can reverse dysbiosis with this. You can reverse type B malnutrition with this. We're up, as I said, to about 1.5 million now. We're operating in over 125 countries. What we're seeing is that when people go into this program, in the majority of cases, where they have symptoms, within about 4 months, we start to see the intensity and frequency of the symptoms of their chronic conditions start to subside. Within 12 months, the bulk of this group are pharmaceutically independent. That's a huge, huge claim to make, and huge claims need a lot of evidence. Here we have a catch-22. We see this consistently in patient after patient after patient. And because we're working with so many physicians who are seeing this in their patients, increasingly we're seeing medical groups and hospitals incorporating this regime into their routine practices, sometimes for patients, sometimes for their high-value personnel who they want to keep well and motivated. I used to work as a regulator. I used to work for the Committee on the Safety of Medicines, which is one of the groups that subsequently became EFSA. So I'm very familiar with the regulatory climate. And what I know is that it's become actually very difficult to talk truthfully and openly about the scientific relevance of the work that has been done in these areas. If we start to say, here's a regime that will cure heart disease, and we can talk about cure now because with these tools, we see reversal of chronic degenerative conditions, then we're going to get blown out of the water. I'll end up in jail or shot or worse. I'll make one more point here. I think what I have been forced to reconsider is the very nature and definition of chronic degenerative disease. We have classically thought of these as entropic conditions, which cannot be cured. All we can do is manage them as the patient's condition continues generally to gently decline. Because we have focused on pharmaceutical tools, which are not possibly curative, because they're specific, they're based on originally Pasteurian concepts. We have then decided, we've made a hugely problematic philosophical error. We have assumed that because these conditions cannot be cured or reversed with the tools we use, they cannot be cured or reversed at all. That's medical nihilism. If you remove these intoxicating factors from the environment, the body's own ability to heal and repair itself is allowed to come to the fore. We see the conditions curing. For example, I have a number of patients now who have been presented to us with evidence of severe atheroma in, for example, coronary arteries or carotid arteries. They're scheduled to go in for surgery. They use the program first, they go into surgery, perhaps we have long waiting lists in my part of the world. 4 to 6 months later, they're scanned again because the surgeons understandably want to know where they're going to work and what they're going to do. And they're then rejected. They're thrown out of surgery because they no longer need it. We see atheroma shrinking. You can do progressive serial angiograms, and you can see that. We know that already you can do that using lipophoresis, but that's invasive and it's expensive and it's time consuming. So we know that theoretically this is possible. We achieve the same result using this type of protocol. We see BMD increasing even in postmenopausal women. We see intra-articular space increasing in people who have osteoarthritis. I'll take a step back because there's a lot to cover here. It's important to try to get to these conditions early because if they've progressed to a certain point and there's been a substantial and significant loss of functional tissue, you get to a point of irreversibility. There is an event horizon. So there is a huge importance in addressing these issues early and, if possible, optimally at a pre-symptomatic, pre-clinical phase, if you can.
Right. Yeah, I think Richard has a question.
Right.
Well, first of all, that was one of the best lectures I've heard in a long time. Thank you very much. It was very interesting, and I didn't take enough notes, so I'll have to go to your blog and get some more information. I'm a
Thank you,
nephrologist.
Richard.
I'm a
Right.
nephrologist. I actually did 3 months of my medical school clerkships in the beautiful city of Edinburgh. So
Oh,
such a
a
great,
fellow alum.
that was so wonderful. One of the three
Mhm.
best months of my life there.
Right.
Incredible. A couple of things. One is I was born in 1950, so I remember those days still, and people were mostly slim. I did, see a rise in vascular disease. I'm totally with you with the changes in the diet being the major trigger. And I've identified really the seed oil consumption as being the biggest factor that was pushed by the dietary guidelines that started to take effect around 1980, which told people get off of the traditional saturated fats like butter and lard and take Crisco and canola oil instead.
Mhm, mhm,
To
mhm.
me, this was the major thing that led our health in a downward spiral, among other things. And of course, the pharma model is so flawed. And at a certain point, it became dominated by evidence-based medicine and guidelines, which then became prescriptive. They
Yeah.
told doctors how to practice so that everybody with a certain lipid number would have to go on a statin, for example. And these statins are extremely harmful in my view, and they don't get at the root cause of the problem. Cholesterol is not the problem. So just a question, um, the effect of smoking also. I mean, certainly in the post-war period, there was a huge increase in the population smoking cigarettes, and that clearly led to, I think, the heart disease epidemic that was eventually blamed on fat by Ansel Keys, right? Erroneously, it was mostly the smoking and big increases in sugar intake. And of course, seed oils were starting to be introduced into the diet back then.
Richard, you've raised so many really excellent points, and you've expressed them very elegantly. And I think that if we had listened to John Yudkin as opposed to Ansel Keys, I think that things would have taken a different turn. Can I just say that we are the same vintage? 1950, it was a kid, it was my birth year as well. And like you, I've seen this incredible transformation in body shape. And the tragedy is that this has happened even in those parts of the world where we have traditionally believed people had the best diet and the best health. So in the 50s, the first time I went down to the Mediterranean area, and I was messing around with my parents in France and parts of southern Greece, people were slim, really
Yeah.
slim. And life expectancy there, if you look at, for example, the blue zones, some of which I've been to, um, people could expect up to 7, even 8 decades of healthy life. And that was an average. According to the WHO, the Mediterranean diet no longer exists. It's been steamrolled by the multinationals. The juggernaut has moved from north to south, from west to east, and the Mediterranean diet has gone altogether. And by the way, there was never one Mediterranean diet. There was a range of different Mediterranean diets. And those people who are eating the healthiest diet now are those who are eating the new Nordic diet, which, when you look at its nutritional content, is actually not that dissimilar to the Mediterranean diet. So these are, young hipster kids who are going out and foraging and eating game and berries and vegetables in season for all kinds of reasons. And they are looking from a metabolomic point of view, quite different from the wider European or North American culture. Um, I'm sorry, I'm rambling. Did
No,
I
no,
answer all of your questions?
when I was in medical school in Belgium in the
Right,
70s,
right.
it was a great thing to go and buy a cornet of frites on the sidewalk that were all fried in tallow,
Yep.
right? Double and triple fried in tallow. Well, I was recently back there like a year ago, and about half of the places are using seed oils now. You had to
Yep.
ask before you get them in Belgium, what are you what are you frying your frites in? I mean, it's pretty depressing.
Yes, it is. And it tasted a lot better
Oh,
when they used tallow.
it's unbelievably much better. I mean, it's just incredible. And of course, McDonald's used to use tallow.
Yep, yep, yep. And you have a higher smoke point, which meant that the texture of the frites was better as well. Um, no, I couldn't agree with you more. If you go back to the 19th century, that was a population in England, the mid-Victorian era. People were consuming considerable amounts of saturated fat and cholesterol, by the way. The English were known to the French as les roast beef because they consumed so much beef. Um, but heart disease at that time was only at about 10% of the figures that we see today in a population that had a life expectancy at age 5 that matched ours almost precisely. So I've written, this is an area and a period that I've studied intensively and published a series of papers on. We are suffering tremendously because we have forgotten the nutritional wisdom of our great-great-grandparents. And I think that it really is very important now, particularly when you look at if you consider the Barker hypothesis, if you look at the epigenetic changes that we're seeing manifesting in children now, and undoubtedly their children as well, we're going down a Gadarene slope. I believe we have to pull back very urgently. And I don't have a dog in the political fight in North America. I can't vote. I'm not making a partisan point here, but I do think that some of the things that Bobby Kennedy and the MAHA team are doing seem to me to be a move in the right direction. I won't
Yeah,
say all of them, but some of them.
sure, absolutely. Just
Mhm.
quickly, what are sources of polyphenols and prebiotic fibers that you would recommend?
Okay, well, the different polyphenols have slightly different pharmacological and pharmacokinetic properties. So if you want to establish an anti-inflammatory polyphenolic umbrella in the different compartments in the body, you could do rather well by the spices are probably your most concentrated sources of polyphenols. So people know about turmeric, but actually, most of the spices, cardamom and paprika, I mean, the list goes on and on, that all of them tend to be pretty heavy sources of polyphenols. And by the way, that is the reason why in India, where 63 ratios are extremely high because there's no cold water, so no cold water fish, you can be a healthy Hindu centenarian if you're eating a traditional diet because they're consuming so much polyphenols. But these molecules are considered to be alien, they're foreign by the body, so they're broken down quite rapidly and excreted. So you have to eat them all the time. Whereas the omega-3s, if you're consuming those, we integrate them into our tissues, and the timing isn't quite so much of an issue. Um, I think that one of the problems that we have here in the United States is the misallocation of government subsidies, which makes it possible to buy ultra-processed foods. Uh, they're actually less expensive than buying the basic foods. And that is a reflection of this misallocation of subsidies. I think that the administration should look at this issue critically and start to consider putting more subsidies into the production, not so much of calories and meat and grains, but into orchards, into vegetables and plant and fruit production. Uh, I think that would be beneficial for American health, and it would also be beneficial if the food multinationals could be persuaded or forced to assume a more health positive position. At the moment, they privatize the profits from the foods that they make and they socialize the costs, which we bear. They're on our shoulders, on the shoulders of our patients. And this, in the end, I think you could say that this becomes somewhat of a political problem. Health is a culturally embedded and therefore politically embedded process. And at a certain point, I think we need to talk to Washington, or in my country, to Westminster, and talk about ways in which the national diet could be improved and how it could be made easier for people to have access to um, to basic produce.
Big food and big pharma have made so much money that they have captured the regulatory agencies. And for example, these seed oils, were never really tested for human consumption. I mean, they were GRAS, generally recognized as safe. They were never formally tested. And I think
Yeah.
Kennedy is talking about having them tested.
You know who put him up
Crazy.
to that? That's Artemis Sinopoulos. So she's been an advisor to a number of these groups, and I have to say that she was one of the people who first got me interested in all of this. But there's many other extremely good scientists who've been involved, and if I'm doing anything at all of any good, it's because I'm standing on their shoulders.
Thanks very much, and I really appreciated the talk.
Thank you, very kind of you.
Okay. Anybody else have Victor, you have your hand up?
Yeah. Yeah, fantastic lecture. I really appreciate it, Dr. Clayton.
Thank you.
One thing that I see that you may have skirted this issue on purpose. I'm not sure, but you mentioned microplastics as a contaminant, food contaminant that's causing disease. I mean, there's so many other things, and I think the number one on the list is glyphosate, and you didn't mention
Yep.
glyphosate, and I'm sure that that's something you can talk about. And the second thing that I think it was a pet peeve of mine for about 20 years is aspartame. Aspartame was one of the biggest pharmaceutical overtakes of our consumer diet in the United States. And I've known for 20 years that it's a neurotoxin. And all of a sudden, a month ago, a research article came out. I don't have the exact citation right now, but they're saying that they didn't even mention aspartame. They said artificial sweeteners are implicated in early neurodegenerative disorders. And then you read in the article, of course, it's aspartame. And I mean, this is something that has been going on for a long time. And I just wanted you to comment because I know you have a lot to say. That's all.
Victor, that's one of those tripwire issues. So with this particular paper you're referring to, was this a sequential study, or were they simply documenting an association in a given population? And if so,
Yeah.
were they able to say that this was an association that was causative or merely?
Yeah, I think this was a epidemiological study over 15 years or 10 years maybe. But all I'm saying is that I've never seen anything like that come out. But finally now the truth rises to the surface eventually. And I believe in Europe, they don't use as much aspartame as they do in the United States. And that's the other thing too, is Italy, many other countries used to be safe from glyphosate, but because Bayer bought out Monsanto, now Europe is prey to all of these things. So and that's why you're here, because you know the truth.
I won't argue with you about the potential problems of glyphosate. I am alarmed, as I think you are. I want to come back to this neurotoxicity issue. One of the original stories that kicked off the aspartame neurotoxic story was the apparent coincidence when aspartame was released in almost the same year, there was a spike in brain cancer. I don't know if you remember that. And the cancers continued to go up. I got to say that I don't know any carcinogen so potent that it's going to show up in clinically demonstrable cases within 12 months. So I think that the fact that these two issues happened at around about the same time actually rules aspartame out as a significant cause of brain cancer. People have sweet tooth. They have sweet teeth, I should say. And if you have an alternative between an intense sweetener and sugar, I mean, you're between the devil and the deep blue sea. Sugar is manifestly harmful if you're eating it in large doses and you're not sufficiently physically active. That leads to metabolic skewing, which now in the latest data that I've seen affects 93% of all United States citizens, all men, women, and children. Now that's really very serious because you're moving towards kidney disease, liver disease, cardiovascular disease, and all of the rest of the long-term problems that come along with metabolic syndrome and subsequently type 2 diabetes. Sugar, yeah, something we really know that there are problems associated with. When you come to the intense sweeteners, first of all, I don't think we can lump all these together because if you look at the different molecules, they're completely different. They clearly have different pharmacological attributes in the body. So saccharin is not the same kind of molecule as aspartame, and that's not the same as the steviozides. These are all very different things. Miraculin is a protein, for example, that's acting on your sweetness receptors as well, but it clearly is doing other things. The most convincing data I've seen for something like sucralose is that it may induce a minor cephalic insulin response. But if you try to look for connections between sugar sweetened beverage consumption and intense sweetener equivalents and subsequent risk of various disease states, I got to say it's the sugar sweetened beverages that keep on coming out in front. So it, it's not as if we're saying to people, here's something you can do that's completely safe and here's something that's risky. I think we have to talk about degrees of risk. And from where I'm sitting, I think the intense sweeteners look like a less harmful way to go. I mean, the thing is with the sugar sweetened beverage and sugar in general, the link with MASLD, MASH, and then you're going down towards the liver cancers is so clear. It's so very clear now. And the mechanism of action is understood. With the sweeteners, the picture is just much more diffuse. Could I add to that, Victor? And I don't want to take up all the time. When I was working for the Committee on the Safety of Medicines, I was actually one of the scientists responsible for looking at the aspartame data. And I was handed the entire dossier, which was a pile of files, which was about 8 feet tall, literally. And it took me about 2 weeks to go through the entire set of files. And I when you have that much data, you'll see something that looks a little bit significant here and a little bit significant there, but I could not see any clear indication of harm or any clear trend. And that was my recommendation. My analogs in other regulatory agencies in different parts of the world came to pretty much the same conclusion. Now, I know that there is a kind of nutri-puritanism, let me put it that way, and people think you shouldn't be able to have sweet stuff and get away with it. If you enjoy it, it should be harmful. I'm just not convinced. I've looked through the data this way and that way, and I'm not convinced. There may be something. I'm open to that, but I haven't seen it yet.
Yeah, if I pull out this article, I'll try to forward it over to you.
Okay, please do. I'd love to see it.
Is your email on the website?
No, I'll give it to you now.
Okay, yeah, if you could post it in the chat, that'd be great.
Okay, let me do that. I'll put it in the chat. Because, yeah, I definitely want to see this paper.
Yeah, and I think it's very similar to the other things we're talking about. We're not talking about cause and effect that's short term. This is long term degenerative processes that take studies that look at long term chronic disease over a long period of time. And I once had a patient that if they had half a can of Diet Coke with aspartame, they were so sensitive to it that literally they would have to be put on a respirator in the emergency room because they'd go into respiratory arrest. And I mean, obviously that's a genetic thing, and it's just anecdotal, but I've seen some people that are very sensitive to it. It's a chemical, and I'm with you. We have to go back to old school. My grandmother grew up in Italy, and she lived in Morocco till she was 85 years old. And she lived to 105 years old. She was born at the turn of the century. And if I could just go back to those days where you could pick dandelion greens in a field without eating all the chemicals and buying pasta and our bread, I mean, bread is a big deal. Buying a loaf of bread without getting contaminated with poison. We can't do that anymore. We can't take that for granted. And I'm with you. We have to go back to natural. And the diets and the food companies have forced us to become addicted to sweet food, and that's the real issue. Why can't you have a can of seltzer with a tiny little bit of flavor to it and be satisfied? But you have to have something that's sweet and addictive. It's literally food addiction, and I'm with you 100%, and I'm behind you in all your efforts.
Again, you've touched on something very central, this issue of food addiction. And the food companies use something called bliss point research to create addictive foods. Are you familiar with bliss point research? Okay, well, it used to be back in the day, if a food company was going to develop a new product, they might have a dozen different variants, and they give it to an audience, and you've got someone with a clipboard walking around saying which ones you like best. You add up all the scores, and at the end of the day, the product, the variant with the highest score is the one that goes into production. It doesn't work. People, they forget, they tell you what you think you want to know, they lie, it doesn't work. So what the food companies do now is they'll give you these different samples while your head is in an MRI scanner. And they are measuring the activity in the pleasure centers in various parts of your brain. And at that point, you cannot hide. They can tell exactly what it is that you like. And they can then manipulate the levels of sugar, fat, various textures, crunch, for example, until it reaches the maximum excitation of the pleasure centers. And this is how they have succeeded in creating a generation of ultra-processed foods which are hyper-palatable to the point of showing all the criteria of addictive substances. And I can share with you that there are clinics that I'm aware of that actually now treat food addiction, ultra-processed food addiction, with naloxone. And they're getting good results. So yes, I think that this is again indicative of the lack of morality of the large food companies. And the problem is, as you say, they have, along with the pharmaceutical industry, they have gone a long way towards co-opting the regulatory agencies, which I do not believe serve the interests of public health in the way that they were originally designed to do.
You mean Frosted Flakes aren't great?
All right, it's Aaron's turn.
Dr. Marinucci
Hey,
has his hand
thanks,
up.
thanks, Dr. Klasner. It's nice to see your face, my friend. Hey, Dr. Paul, out of all the things that you could have done, why did you choose to sit on the scientific advisory board of Zenzino?
Okay, I'll tell you why. I was having a really good time at Oxford, good conversations in the Commons room, and the finest selection of wines known to man. They had been working on their wine cellars for the last 1,000 years. So I didn't want to leave, but at a certain I didn't want to go. But at a certain point, the science that was emerging not only from Oxford, and I was working with some brilliant scientists there, but in many other centers all over the world, had become so, I think, convincing that actually it became a moral imperative for me to leave the university and get my hands dirty. And because of the way in which the regulatory system works, the only way that I could get a soapbox to stand on and start talking to people was within the context of a direct marketing organization. If it was a retail organization, you were so constrained in terms of what you can say. You can't really tell the truth. You can't say, well, okay, there's a study that shows this, and I'm pretty certain that it means that if you do this, there will be some sort of outcome. When we're talking together like this in relatively small groups to people that we know, friends, family, or etc., we can talk openly about our personal experiences. I can talk openly about the science that I've looked at. And it's just an environment where it's like a samizdat. For those of you who remember how things operated in the Soviet Union, when all the media outlets are controlled, when even the typewriters had signs in the keyboard, which meant that any letter could subsequently be traced back to an individual typewriter. What they did was they would print up single sheets of paper and stick them on the walls of abandoned buildings. That was the samizdat. As far as I'm concerned, a company like Zenzino is the modern day and widely disseminated version of a samizdat. I can give lectures, I can write, posts in my blog, and it's going out under the radar by and large. So it just means that I want to shift the needle when it comes to public health. And it seemed to me that was the most effective way of doing it. One of my
Okay.
heroes is Jon Snow, not the Game of Thrones guy, but the father of epidemiology. And he was working in the 18th, 19th centuries at a time when the major causes of disease and death were waterborne plagues, cholera, typhus, typhoid. And at a certain point, God knows how, he has this map of London on his desk and he puts pins in all the different houses where deaths are being reported. And at some point, he realizes that they're clustered around a particular well. And the famous story is in the dead of night, he and his manservant go down to the well and they remove the pump handle. In 2 days, the outbreak is finished. It's gone. And I feel that I'm following a long way behind in the footsteps of Jon Snow. The academic stuff was great, but it got to the point where I had to go and try and take the handle off the pump. And
Love it.
Zinzino gave me an opportunity to do something like that.
That's fantastic. Dr. Klausner, we have the Benzino. We have that diet that your grandma used to have. So we have the Mediterranean diet in a bottle. Just
Okay, great.
if you're interested, just talk to Austin. He'll take care of you. Okay.
Perfect. I'm down.
All right, my man. Appreciate it. Any of you guys? Well, I don't know if I'm allowed to, but Doc, it's okay to kind of plug this a little bit. Anyone that wants to get their test and see where they are with their ratios on their omega 6s and 3s, go to testdon'tguess.info, and we can take care of that for you. It's a blood spot test, first of its kind. In about 20 days, we'll have your number for you, and we can help you get to where you need to be so we can get rid of this chronic illness that's going on.
Aren't my 20 days up yet, Aaron?
Yeah, actually, no. You got probably another week. We just did it last week. There, Doc.
It drags on so slow.
We're about 10 days out, Doc. We'll have that info
right.
for you.
Okay, every day I wake up saying, when is it going to be ready?
Thank you, Doc. Thank you, guys.
Okay. Thank you, Aaron. Thank you, Dr. Clayton. Thank you, Alan. This was fabulous as always. We will have it up on our website in a 24 to 48 hours, hopefully, if I remember to do it. And it will be probably we'll be able to make this one on YouTube also. So it's Clearfield Medical Group on YouTube. The website is aosrd.org and /webinars. All of our programs are there. I don't have a speaker for next week, so if anybody wants to volunteer or knows somebody, let me know. Even that we will have to drag Dr. Clayton back. So, okay. So, any other questions? Dr. Nario, do you have anything online?
I think on the chat there's something. John has a question. Pop up.
Something about stevia, right?
Yeah.
Replace sugars.
Yeah, we didn't mention that, but we talk about the bad of sugar and aspartame. Can you replace both of those with stevia? Is it safe?
The safety data that I've seen on stevia looks perfectly adequate. I would just point out that however you purify it, you still have some of those licorice notes, which you might like, but I don't. There's alternative sweeteners that you can use. Monk's fruit, I think, has a profile that's a little closer to sucrose, but in the end, it's all accounting. It's a matter of taste.
Thank you.
All right. Okay, I think that was the one question we didn't get to. And I guess testdon'tguess.info. Right, and Zinzino balance test, of which I'm waiting for my answer. I'm sure I'm going to be way off on the side of no good. I fail all of these tests. So
hey Bill,
yeah,
were you able to get a hold of the cardiologist Sam Fillingane?
No.
Okay.
Send me a contact.
I'll text it to you. He was willing to do the
all right
meeting.
Say no more.
All right.
Oh, thank you very much.
Okay, thank you, Dr. Clayton. Thank you, Dr. Alan, and Aaron, wherever you are driving. Dr. Nario, great as always. Thank you so much. We'll see you next week, same time, same station. I know A4M is coming up.
April
Next week I'll be in Miami for American Academy of Stem Cell Physicians, March 20th to the 22nd. It's in Miami. It's so you can look them up if you want to join them. We always have some fun there. Um, and then AMMG also, April 15th to the 19th. We'll be doing some fun things there too. So those are some of the meetings coming up. I think you have something online coming up also, don't you, Dr.
Oh,
Nario?
we're going now. This is our women's hormones for APRAM.
Okay. All right. When does that start?
Oh, it started yesterday. So we have quite a few, a lot of students online. And
Okay.
that reminds me to check test papers again. So thanks for
No,
reminding me that about that.
no worries. And you want to can you let us know again about the CME credits for the homeopathic
Oh yeah, so
stuff?
we are now accredited for homeopathic units, CME units for every episode of our Tuesday webinars here in AOSIM. And we are now also going to be bringing in some of the pure homeopaths into the fold, as I mentioned, Dr. Clearfield, because they will definitely want to try to get the units that we offer here. So yeah, we're going to be meeting more people, like-minded people, and we're going to interact with them as much as
Yeah.
we can.
And like I say, after 45 years, I finally got an MD degree. That's right.
There you go.
My mother's so proud. It's an HMD degree, but I got an MD degree finally. So,
Hey, it's always meant to happen.
so there are some advantages. The Homeopathic Board of Nevada covers pretty much all alternative, what we call alternative medicines. They'll back you when you're stodgy old board will wants to put you out of business. So there are some advantages. Do I have that right?
Yeah, so one of the things that we are getting, so the Andrew Weil program, A4M fellows are now jumping in for the privilege of actually doing telemedicine consult by applying for the Nevada Homeopathic Board licensure. So that's going to be a privilege that we actually will cover. So all of us are integrated practitioners. The difference between you applying through the medical board here is, of course, you're not going to be covering dietary supplementation, I guess, consults, all those integrative consultations that you will have. So that's going to be related now to the insurance carrier that you will have. So with the integrative medicine practice, the Nevada Homeopathic Board will definitely grant these licenses. Even if you only choose to be just online consultation, you don't have to be here. And that's going to be something that I guess is enticing a lot of practitioners to jump in. So we invite you over to apply for that. If you're interested, just email us, me, Dr. Quinn here, and Dr. Clearfield. So we're all part of the Homeopathic Medical Board here in Nevada.
So that's right. And the executive director is
Christy Steele, Dr. Bill's
has been my
left,
right hand person for almost 10 years now. So it's a close shop. So,
your life rate hand lady.
yes, yes, I wouldn't be able to operate without her. So okay. If you have any questions about that, you can direct it to me, Dr. Nario. He seems to have taken the lead there. Any last words, is Dr. Clayton still on, or did he leave? Any last words? Any sum up?
My question is, Dr. Nadio, which one is your lecture that you for hormones? It's a4m.com, but what is the name of it?
It's actually the women's hormones. I don't know if the link is still open for registration because we started it already. I'm gonna send that to I have your email. I'll send you a link if I can find it. But for sure, spring Congress. That's going to be a good one in Florida.
Yes. Okay.
Okay. Bill, do I get a last word? I'll be brief.
You get the last word.
Okay. I'll just say one thing. The evidence that we have from the 19th century and from the other vestigial groups is that if we can get the nutritional status right, it looks very much as if we can reduce the burden of chronic degenerative disease by about 80 to 90%. In those populations, the levels of disease are about 10% of what we see here. And that matches the answer that you'll get if you ask a cardiologist or an oncologist, how many of your patients have strong genetic risk factors? About 10%. So we think when we look at those populations that the only people who are developing these conditions are those individuals who carry those genetic risk factors. Now, if you extrapolate that to the modern American situation, a 60% market penetration of this type of approach, you'd be saving, and I've had some good med-economists look at this, you'd be saving about $4 trillion of healthcare costs a year. And at that point, you're not only paying off, the national deficit, you're actually paying down the national debt. Right? That's pretty significant money. And so we're imagining a really very different configuration of public health care if we can get to it.
Okay. All right. Okay. And Alan, you got anything for us?
Just want to thank you so much for the opportunity to be able to share tonight. And Dr. Clayton, thank you so much. I owe you a big one.
Yeah, thank you so much, Dr. Clayton, and Aaron, wherever you are out there in the TV land. Thank you also.
California, baby.
Okay, there you go. Yep, 10 miles from Reno. Okay, in other words.
Yeah, that's right. But hotter, but warmer.
It's been pretty nice here all the last week or so.
Oh yeah, cold plunged the river yesterday.
It's it's been spring here already. So okay, good night, everybody. Thank you so much.
Thank you again.
Let us know let us know where and when you are, and if you're interested in any topics, if you want to present, please let me know. Okay. Thanks a lot. Good night.
Thank you.